So this exercise is related to topic alcohols and the halogen alkanes. In this question, in the human body, one chemical have a number of different effects. An example is there, retinol, one of the substance in vitamin A. The structure is shown here, and it can be converted into retinal. Look, retinol can be converted into retinal. So this give an idea. It's an alcohol which can be converted into an aldehyde. And a form of this combined with opsin molecule in the retina of an eye to make a light sensitive pigment. When the light enter the eye, the shape of retinal molecule is changed, and which result in a separating from ospin molecule, and the outcome of this separation, we see a light. That's actually, there's a photochemical reaction which happen in our eye, a reaction which is initiated by a light. We call that as a photochemical reaction. So because of this photochemical reaction, we are able to see. The retinol can also be converted into retinoic acid, which is an important in the body for the correct maintenance of a mucous membrane. And the failure to this to produce retinoic acid can leads to a condition known as a dry eye, which can also result to a blindness. So basically, what is happening? It's the retinol when a light retinol is converting to retinal, and then back because of the light. It's the main thing is there. Light sensitive pigment is there. So the function of this vitamin and other nutrients illustrated need for a healthy and a balanced diet. So the carrot, that's why, like example, carrot is having a high percentage of this chemical. So we, for the eye, it is beneficial. Or any food which is having enriched in a vitamin A. So retinol has an alcohol functional group. Classify the type of alcohol group in retinol and explain the meaning of the term functional group. So which alcohol is this? Like the type of alcohol, is it a primary, secondary, tertiary? What kind of alcohol is this? So if you want to check whether it's a primary, secondary or tertiary, you just have to check how many carbon atoms or how many carbon chains are around. So this is our OH. So this is our OH. So this whole group is one. Like this carbon which is having two hydrogen and rest all is like when I count from here, I'll count from this one. So only one is there. So when one is there, it means it is a primary the alcohol. And what is the meaning of the term functional group? Functional groups are atoms or a group of atoms which can chemically change the properties of a substance. We call that as a functional group. So the part of a molecule which react or a group which is responsible for the distinctive characteristic, we call that as a functional group. Next, retinol can be oxidized to an aldehyde retinal. So remember, alcohols can be oxidized to aldehyde, and aldehyde can further oxidize to a carboxylic acid. If you use a vertical or a reflux condenser, then what will happen? There will be a complete oxidation that will result in a formation of a carboxylic acid. But if it is a partial oxidation, that will form an aldehyde. To illustrate the conversion of an alcohol to an aldehyde in the lab, a student suggested using the following apparatus and an excess of an oxidizing agent. Explain why this proposed method would have been unsuitable for the production of an aldehyde, and explain what modifications are necessary for a useful conversion. A new diagram is not required. You just have to mention, because this is a reflux condenser. What is the purpose of the reflux condenser? The purpose of a reflux condenser is if we want a complete oxidation, then we use a reflux condenser. Here we don't want a reflux condenser, so the reflux apparatus will produce carboxylic acid or retinoic acid. So what we have to do, we have to convert this into distillation. Like we don't have to draw that. We just have to change the position of a condenser. The condenser should be in a horizontal position rather than a vertical position. And the oxidizing agent should not be in excess, because excess of oxidizing agent will be used if we want a carboxylic acid. So we should not use an excess. We should only use a limited quantity of a limited quantity of a oxidizing agent. So the 3 points we'll mention. One is this is a reflux apparatus which will result in a formation of retinoic acid. So we have to change the position of a like convert that into distillation or change the position of a condenser and use a limited amount of oxidizing agent. Is it clear? Then we have the oxidizing agent suggested was a sodium dichromate in acidic solution. Complete the ionic half equation. Remember, whenever we are balancing an equation, if you want to balance the oxygen, you add water. If you want to balance the hydrogen, you add protons, and then you balance the charge by electrons. Okay. So first Cr is 2 here, so I should put 2 because the chromium should be balanced. Then oxygen is 7. So how many water molecules do we need? We need 7 water molecules to balance this equation. What about hydrogen? 7 multiplied by 2 makes 14. So we need 14 hydrogen. So there will be 14 protons. And then what about the net charge? You have to check the left and the right hand side. So here the net charge on this molecular ion is negative, and the net charge is plus 14. So the total charge is plus 12. What about the net charge? It is plus 6 here. This water is neutral. So we want the net charge to be like plus 6 on this side, right hand side. So we need plus 6 on the left hand side as well. So how many electrons do we put? We will put 6 electrons, so that the charge, because adding 6 electron means it's negative 6. So as a result, what we'll have positive 6 here. Then the oxidation numbers, the oxidation number of a chromium in a Cr2O7 with a charge negative 2. So it is Cr2O7 with a charge 2 minus, each oxygen is minus 2. So 7 will be negative 14. So the chromium should be plus 12 total. But for 1, because 2 are plus 12, so 1 will be plus 6. So here the oxidation number should be plus 6. And here what the charge on the ion is same as the oxidation number. So oxidation number will be plus 3. Look, there is a difference when you write the valency, you write number first and the charge after. Like example, you write 2 minus 3 plus. But when you write the oxidation number, it should be the charge first, it should be the nature first, like positive or a negative, and then the number afterwards. So when you mention a valency on the ion, you mention the number and then the nature. But when you write the oxidation number, it's the nature first, like positive or negative, and the number afterwards. That's why we wrote plus 6 and plus 3. But here, when I am writing, I should write as 2 minus, like the charge here in the chromium. I mean dichromate. It's written 2. So these are the valencies, and these are the oxidation number. So for valency number, then the charge, like I mean positive or negative. But for oxidation number, positive or negative first, and then the number. The color you have to memorize that the chromate, when it is plus 6, it is orange in color, and it is plus 3, it is green in color. So you have to memorize this. And same thing for manganate, like manganese plus 7 purple or pink acceptable, and plus 2 is colorless. The next is describe 2 features in the infrared spectrum which could be used to determine whether retinol has been completely converted into retinal. So the retinol will have COH bonds will be there, but in retinal will have C double bonded with O and H bonds will be there. So how we can identify that the retinol, because when an alcohol is converting into aldehyde, we just have to distinguish what are the changes in the infrared spectrum, like the absorption spectrum, infrared spectrum will be there. So in retinol, because there will be C double bonded with O, so C double bonded with O of aldehyde or a ketone. So this peak will be there, 1740 to 1720 in this retinol. But for OH, so when initially what will happen, we'll have this peak in the molecule, then the two peaks both will appear. Why two peaks will appear? Because not all the molecules instantly will convert into an aldehyde. So the amount, like this peak will disappear. First we'll have OH, then during a reaction we'll have both OH and CO, and once it has completely converted into retinol, it will have only C double bonded with O. So that's how you will mention this. Is it clear? So in retinol, there will be a strong absorption from 1740 to 1720. But in retinol, there was an absorption of the broad absorption was there, and eventually it will disappear. What are the two features exactly what you have? The two features what you have to mention. Number one, that initially we will have the peak. Initially, we'll have the peak because it's an alcohol, we'll have a peak from 1750 to 3200. During a reaction, the another peak will appear from 1740 to 1720. And then once the reaction has completed, this peak will disappear. Why this will disappear? Because alcohol has been converted. So we won't have a peak of OH, we'll have only peak of C double bonded with O. So you should relate, like the answer should be structured answer. Like you relate your points one to the next. First, in the beginning, OH absorption is there. Then as a reaction start, the aldehyde start to form. So it will have a peak due to OH, which is already there, and due to aldehyde, which is formed. And once the reaction has complete, there is no peak because of the OH, and only peaks will get due to C double bonded with O of aldehydes. In part 4, the structure of a retinol molecule that combine with opsin in the human body differ from a structure expected from oxidation of retinol. The structure of retinol and retinol are shown, as well as the oxidation structure, changes, as well as in oxidation, structure change has occurred within the molecule. Circle only the part of a retinol molecule where the change has occurred. So we have to circle which part the change has happened or occurred. If you compare the molecules, the original molecules are there, because if you see the first structure, it's actually a geometrical isomer. If you see C double bonded, because here is a carbon-carbon double bond, here heavy groups are attached up and here heavy group is attached down. So this was a trans. But as you can see here, this part, it is attached up. If I compare, like when we compare the changes, there's no change till here. This is same. There's no change here as well. What about this one? This one has changed. Like this part, the H is up. Like this group has moved up. Here, this group was moved down. Like actually, when heavy groups are on the same side, so this will be a cis. Like this was a geometrical isomer which was cis because heavy groups are on the same side. And or we can say E. Uh, Cis or Z, I mean not E, E is a trans. And here we have a C double bonded with C. So this group is attached up, the other group is attached down. So we just have to circle the part, so as circle on the part of the retinal molecule which where this change has occurred. So we know alcohol has converted into an aldehyde. How retinol is an alcohol and retinal is an aldehyde. No, that what they are saying, that it is the molecule. This is a retinol is there, and then it is converted into retinal, but the geometry of the molecule might be changed as well. That is what is mentioned here in the question. That the structure of a retinal molecule that combined with opsin, in the human body differ from a structure expected to form from retinol. So what is the reason why there is a difference? What difference we observe? It's actually it's a geometrical isomer. Like this is a carbon-carbon double bond, carbon-carbon double bond. So the molecule has been twisted. So we just have to circle that part where the change has happened or occurred. So where the change has happened or occurred, like this is the region where the change has happened. Is it clear? Which is trans? How to identify which is trans and which is E? When you check, this will have hydrogens here. So heavy groups are attached on the same bottom here. So this is the same side, cis. But when you check for this one, hydrogen is here and another hydrogen here. So one group is attached up, another one is attached down. So what is this? This will be a trans. So this will be a trans or E, and this is cis or Z. The bond angle around each carbon atoms, and which part do we exactly circle? I did circle. You just have to circle the part where the change has happened. So you have to circle this part. But we have to circle on retinal. So basically here for a retinal. I circle both parts. So just to give an idea that where the change has happened or occurred. The bond angle around each carbon atom in a carbon-carbon double bond is 120. Explain the reason for this bond angle and state the name of a shape. So bond angle of a carbon, when the bond angle is 120 degrees, so what we call the shape is it's trigonal planar. And whenever they ask why it has a specific shape, so you will always refer to a bond pair and a lone pair. Like according to electron pair repulse, it will be trigonal planar. And the reason is that when we are predicting a geometry, we consider the double bond or a triple bond which are in the same line, we consider them as one. So if there's a double bond or a triple bond, we all consider this as one. So one, two, three. So when we predict the geometry, we'll say there are like three areas of the electron or three regions of the electron density, like one, two, and three regions. And whenever there are three regions of the electron density, the bond pair, so they try to minimize the repulsion. And when they minimize the repulsion, they will have a maximum separation. No, you cannot say one lone pair. There is no lone pair present. There are four bond pairs. These are actually all of them are bond pair, like carbon, carbon, C double bond. They all are bond pair. There are actually four bond pairs, but because when you are predicting a geometry, you will say three regions of the electron density or three regions of the electron pair. Three regions like one, two, three. and they try to minimize to maximize the separation. So that's why they maximize the separation when the bond angle is 120. And this will be trigonal planar. Is it clear, this one? Then we have part E, complete the diagram to show the skeletal formula, complete the diagram, skeletal formula of retinoic acid, and give the observation. Retinoic acid and retinol both have OH group, suggest one chemical test. If you want to distinguish alcohols and the carboxylic, both contain OH groups. So how we can minimize, how we can check? Give a positive observation for the test and state one necessary experimental precaution. The suggest one chemical test that could test the presence of OH. So for OH, we normally add a PCl5, phosphorus pentchloride. An observation will be a misty fumes will be there, and misty fumes HCl is there, so we should use a fume cupboard. For a reaction when it happened, we should always use a fume cupboard. That will be our precaution. And we just have to complete the structure here, making it to a carboxylic acid. So this will be a carboxylic acid, C double bonded with O, and OH will be there. Because that's they mentioned the first part, they ask convert an alcohol to a carboxylic acid, and then how you can test the presence of OH by adding a PCl5, observation misty fumes, and use a fume cupboard.